Good morning all. Today we are going to discuss a new topic, that is microscopy. So in the previous sessions we have discussed about growth, factors affecting the growth and various different methods for the measurement of growth. So now onwards today we will discuss about microscopy. So why do we use microscopes? Actually these microscopes are used to see things, that is objects or organisms which are not visible with our naked eye. And also we are using these microscopes to study the morphological properties, that is external characters of the microorganisms at micro and nanoscale length. Then also to collect images at high resolution and also to observe live phenomena. That is, using microscopes, we can observe live cells, chemical reactions under microscope. And the term microscope, it was derived from the Greek word micron, that is M-I-K-R-O-N. Micron means small and scopios means to look. So microscopy, it is an instrument for viewing objects which are too small to be seen with our naked or unaided eye. Now the microscope is an instrument for viewing objects. So what do you mean by microscopy? It is the science of investigating small objects using such an instrument called microscope. Okay. So these are the different magnification of microscopes. So we can see the image which is visible with our human eye, that is 10 meter, millimeter, 10 millimeter, 1 millimeter, etc. This can be visible with our human eye. And this portion, it is not visible with our human eye. This can be visible through light microscopy. That is human hair, majority of cells such as RBC, WBC, bacteria, etc. And this portion, bottom portion, these are those which are observed, and it starts from 100 micrometer to 0.1 nanometer. These are the region where those objects which are visible through electron microscopy. So these are mainly viruses, macromolecules and atoms. So these are visible through electron microscopy. So electron microscopy can also be used to do bacteria, human hair, etc. But this portion cannot be visible using light microscopy. Now the purpose. The purpose of this microscopy, it is to become familiar with the history and also the diversity of microscope instruments, and also to understand different components of microscope and how to use the microscope and how to care the microscope. Then third purpose, it is to learn the correct use of microscope for observation and measurement of organisms. So these are the main purpose of microscopy. Now coming to the introduction. Microbiology, it is the branch of science that has so vastly extended and expanded our knowledge of living world, owes its existence to Anton van Leeuwenhoek. So in 1673, with the aid of crude microscope which he invented, which consists of biconcave lens enclosed in two metal plates, and this Leeuwenhoek introduced the world to the existence of microbial forms of life. So these are the historical aspects. So we can discuss some of the historical aspects which are dealing with the invention of microscopy. Historical background. Zacharias Janssen, that is, in actually in 1590, Hans Janssen and his son Zacharias Janssen, they both developed the first microscope. So the first microscope was developed by Hans Janssen and his son Zacharias Janssen. And this Zacharias Janssen is a Dutch spectacle maker who tested several lenses in a tube and discovered that nearby objects appear significantly enlarged by viewing through the microscope. Then second person is, which I didn't have mentioned here, that is in 1620 there is another one, Christian Huygens. He was another Dutchman and he developed a simple two-lens ocular system that was chromatically corrected. Later it was chromatically corrected. അപ്പോൾ second scientist എന്ന് പറയപ്പെടുന്നത് 1620 Christian Huygens ആണ്. He was a Dutchman and he developed a simple two-lens ocular system. Then the third person was Robert Hooke in 1665. Robert Hooke. He was an English physicist and he looked at silver of cork through a microscope lens and he noticed some pores or cells. Above this, Robert Hooke used a microscope to observe thin slice of cork. This is the cork tissue which he observed through microscope, and the spaces he saw reminded him of what? Small rooms where monks live. So he called them cells, which he used to describe the smallest units of life. So the cells are the smallest units of life, and he used microscope with 2 and 3 lenses, but they didn't produce very detailed images. So these are the drawings made by Robert Hooke. Then this is the microscope which was invented by Robert Hooke. That is, and he published his findings in Micrographia. That is, this is the first page of Micrographia, and he presented his findings in Royal Society of London at that time. And this is the microscope which he invented. And his time was in between 1635 to 1703, and Micrographia, which he published in 1665. Next, about another important personality, that is Anton van Leeuwenhoek, and he was a Dutch merchant who learned how to grind lenses to make simple microscopes, that is, which consist of only one lens. These produced clearer and more enlarged images than Hooke's. For Robert Hooke in the microscopic image in a compare, the images produced by and discovered by Anton van Leeuwenhoek using one lens system was more clear and enlarged, and he is considered as the father of microscopy and built over 500 different microscopes. And he was the first to discover microorganisms under a microscope by observing a drop of pond water filled with life, and he called them as tiny animalcules. So he first of all discovered the microorganisms observing a drop of pond water. And he also saw and studied bacteria and the blood flow through capillaries in the tail of fish. And actually he was a tradesman of Delft, Holland, and in 1661 he discovered bacteria, free living, and also parasitic microscopic protists, sperm cells, etc. So he was considered as father of microscopy. Then this term microscope, another important historical feature is that which I didn't have mentioned is the term microscope was coined by Faber in 1623. Faber, okay? Faber ആണ് microscopy എന്ന് പറഞ്ഞ term coin ചെയ്തത്. So these are the drawings made by Anton van Leeuwenhoek. Now these are the instruments which early found. Microscope. First compound microscope. Then Leeuwenhoek microscope is this one. Then Hooke's microscope is this one. Italian compound microscope. Culture screw barrel microscope. Early solar microscope. So over the years, microscope has evolved from a simple single lens instrument of Leeuwenhoek with a magnification of 300 to present day electron microscope capable of magnifications greater than 250,000. അപ്പോൾ 300 മാഗ്നിഫിക്കേഷനിൽ നിന്നും ഇത്രയധികം ഡെവലപ്ഡ് ആയിട്ടുണ്ട് മൈക്രോസ്കോപ്സ്. So we can go through it. So the microscopes are designated as either light microscopes or electron microscopes. So the light microscope use visible light or ultraviolet rays to illuminate or observe specimens. So what are the important light microscopes? They include bright field microscope, dark field microscope, phase contrast microscope, and fluorescent instruments. So these are the main important light microscopes. So these fluorescent microscopes use ultraviolet radiations whose wavelength is shorter than that of visible light and are not directly perceptible to human eye. So we are not directly observing it. In the case of this, so next group is electron microscopes. In the electron microscopes, we use what? Electron beams instead of light rays. Light microscope, we use light rays, visible light or UV. But in electron microscopes, electron beams are used. And instead of lenses, glass lens, electron microscope will use magnets instead of lenses to observe submicroscopic particles. Okay. ഇനി നമുക്ക് കോമ്പൗണ്ട് മൈക്രോസ്കോപ്പ്, അതായത് ലൈറ്റ് മൈക്രോസ്കോപ്പിന്റെ ഡിഫറെന്റ് പാർട്സ് എന്തൊക്കെയാ വരുന്നത് ഡിസ്കസ് ചെയ്യാം. സോ ദ മൈക്രോസ്കോപ്സ് യൂസ്ഡ് ഇൻ ക്ലിനിക്കൽ പ്രാക്ടീസ് ആർ ലൈറ്റ് മൈക്രോസ്കോപ്സ്. അപ്പൊ നമ്മൾ ക്ലിനിക്കലി യൂസ് ചെയ്യുന്നത് ലൈറ്റ് മൈക്രോസ്കോപ്സ് ആണ്, കോമൺ ആയിട്ട് യൂസ് ചെയ്യുന്നത്. ദേ ആർ കോൾഡ് light microscopes because they use a beam of light that can be either visible light or UV to view specimens. And actually these light microscopes are mainly simple microscopes and compound microscopes. And in the case of the simple microscope, it consists of a single lens system. And the compound microscope, it is most common microscope used in microbiology. അപ്പോൾ നമ്മുടെ കോളേജിലൊക്കെ കാണുന്നത് കോമ്പൗണ്ട് മൈക്രോസ്കോപ്പ് ആണ്. ഇറ്റ് കൺസിസ്റ്റ് ഓഫ് 2 lens system, that is combination of lenses to magnify the image. അപ്പോൾ ഓരോ ലെൻസിനും എന്തുണ്ട്? They have a different magnifying power. അപ്പോൾ മാഗ്നിഫിക്കേഷൻ എന്തൊക്കെയാന്നുള്ളത് നമ്മൾ പിന്നീട് നോക്കും. A compound microscope with a single eyepiece. ഒരു eyepiece ഉള്ള compound microscopeനാണ് എന്ത് പറയാ? Monocular microscope. The compound light microscope with 2 eyepiece, it is said to be binocular microscope. Binocular microscope. 2 eyepiece ഉള്ളത് binocular ആണ്. Single eyepiece വരുന്നത് monocular. Now this is actually a binocular because it have 2 eyepiece. Okay. So now we can discuss parts of microscope. അപ്പോൾ ഇത് ഷോർട്ട് ആക്കി കൊടുത്തിട്ടുണ്ട്. Main parts മാത്രം. The main parts of microscope are eyepiece, microscope tube, then